Alright, now let's talk about glycolysis regulation and the key enzymes involved. Shown here, we see the glycolysis substrates, intermediates, as well as end products. I'll focus on the important steps, but I first wanted to offer a global picture of glycolysis. Notice how most of these steps have double-headed arrows, meaning that they can occur in either the forward or backward direction. Now what we're going to focus on are the unidirectional arrows, which we can see here. Here we can see the 3 steps of glycolysis that are irreversible, meaning that they only occur in the forward direction. And the net reaction of glycolysis can be shown in this equation. You start with a glucose molecule, you add 2 inorganic phosphates, as well as 2 ADP, as well as 2 NADH+ electron acceptors, and you end up with 2 molecules of each of the following: pyruvate, ATP, NADH, and a hydrogen ion. So what's important here? Well, you use 1 glucose to make 2 pyruvate, 2 ATP, and in this process, 2 NAD+ molecules get reduced to NADH plus a hydrogen ion. Now, if you get confused which species gets reduced or oxidized, don't freak out. Just look at the charge. Here, the NAD+ changes to NADH. So NAD+ loses its positive charge. In other words, its charge is reduced from +1 to 0 or neutral. Or to say this another way, NAD+ is reduced to NADH. Alright, now let's go through the enzymatic steps that are involved to regulate glycolysis. Now the 2 steps in glycolysis that require ATP are the first step and the third step, and you can see these here. This will represent our energy investment phase. First off, hexokinase or glucokinase will use 1 ATP to convert glucose to glucose 6-phosphate. And as we mentioned before, glucose 6-phosphate exerts negative feedback on hexokinase, but not glucokinase. Instead, high levels of fructose 6-phosphate act to inhibit glucokinase. Now, in the third step of glycolysis, phosphofructokinase 1 uses 1 ATP to convert fructose 6-phosphate to fructose 1,6-bisphosphate. And this just means that it's a fructose with a phosphate molecule at the 1 and 6 positions. And while we're on it, do you remember the rate-limiting step of glycolysis? There you go, it's phosphofructokinase 1, which we just talked about, which is the rate-limiting step of glycolysis. Now, since it's the rate-limiting step, there are going to be a lot of things that either activate or inhibit it. Some of these things include adenosine monophosphate, or AMP, and this just means that there's a lot of AMP around. This needs to be converted into ATP, so it's going to activate phosphofructokinase 1. Also, if there's a lot of fructose 2,6-bisphosphate, this indicates that the blood sugar is high, so there's plenty of glucose to be used for glycolysis, both of which are activators of phosphofructokinase 1. Now, if a lot of ATP and citrate are present, this means the cell has a sufficient amount of energy available and does not need to continue in glycolysis. Therefore, ATP and citrate will inhibit phosphofructokinase 1. Now, there are also 2 steps of glycolysis which also produce ATP. Both of these occur twice since they occur after the step where our 6-carbon molecule is split into 2 3-carbon molecules. This means that a total of 2 ATP are initially consumed and a total of 4 ATP are produced per molecule of glucose, resulting in a net gain of 2 ATP. Now that we've already invested some ATP prepping the glucose molecule, at which step do we actually make some ATP? Well, the first step in which glycolysis produces ATP uses the enzyme phosphoglycerate kinase, as you see here. Now, phosphoglycerate kinase converts 1,3-bisphosphoglycerate into 3-phosphoglycerate, and you can see this here. Now, going by the names of the molecules, it makes sense that 1 ATP is produced by this reaction, since 1,3-bisphosphoglycerate, which has 2 phosphates, is being converted into 3-phosphoglycerate, which has 1 phosphate. And remember that this reaction takes place twice, because halfway through glycolysis, glucose is split into 2 3-carbon molecules. And these 2 smaller fragments are called glyceraldehyde 3-phosphate, and they both undergo the same reactions to form 2 molecules of pyruvate. Now, the next enzyme that we need to discuss is pyruvate kinase. And pyruvate kinase is involved in the conversion of phosphoenolpyruvate to pyruvate, and this process also yields an ATP. And once again, as we just stated, this reaction will also occur twice for each glucose molecule fed through glycolysis. Now, what about the activators and inhibitors of pyruvate kinase? Can you think of these? There you go. Fructose 1,6-bisphosphate activates pyruvate kinase, and you can see this here. And if you think about it, this makes sense. If you have plenty of upstream substrate or building blocks for pyruvate, why not activate the enzyme that's going to make it? And on the opposite side of the coin, we see that ATP as well as alanine inhibit pyruvate kinase. And it also makes sense that if you have lots of ATP around, you would not have any reason to make additional ATP just for the sake of making it. But what about alanine? Can you think of why this might inhibit pyruvate kinase? Alanine is converted to pyruvate, and pyruvate can be converted into alanine using the Cow-Hill cycle of ammonia transport by alanine. And if you need a quick refresher, just go check out the Transport of Ammonia by Alanine video. So just like having plenty of ATP available, if you have plenty of a downstream product available, then there's no need to proceed down the pathway just to make excess for the sake of excess. Well, alright, let's take a shot at a quick flash quiz. In glycolysis, which reactions require ATP? Well, there are two steps in glycolysis that require the investment of ATP. And this is converting glucose to glucose 6-phosphate, as you see here, as well as converting fructose 6-phosphate to fructose 1,6-bisphosphate, as you see here. And if you think about it, it's just like investing on Wall Street. You need to initially invest some money, or ATP in this case, before you're ever going to get a return on your investment. And remember, that's just the 4 ATP that you get at the end of glycolysis. And just as a quick reminder, what's the net gain of ATP during glycolysis? There you go, there's a net gain of 2 ATP. Well, alright, that's going to wrap up glycolysis regulation. See you in the next video.